Okay. The DNA genetic material was over by tomorrow. Yesterday. Yesterday these things were over. Today we are going to start with structure of DNA. Okay. DNA structure, and then we'll follow with DNA packaging, how DNA is packed inside the uh, small cell. Okay, and further then we'll come with the RNA. Okay, DNA.
Excuse me, ma'am.
Yes,
Do you remember I asked for
say
you, asked for the pedigree analysis from you?
yes.
Ma'am, in pedigree analysis, we
Let me,
don't only have to search for
beings is 99.9% identical. It is that 1/10 of 1% difference that makes us all unique. DNA derives its name from deoxyribonucleic acid, a type of nucleic acid. Nucleic acids are made up of polynucleotide chains, which are formed by several nucleotides or molecules that make up the structure of the DNA when bonded together. In fact, the length of a DNA is defined by the number of nucleotides or pairs of nucleotides present in the DNA. A pair of nucleotides is also known as a base pair.
okay. Uh, okay.
For example,
Actually, last year in chapter 9, we have learnt about DNA structure in biomolecules. Okay, in that also we have done about that DNA is a genetic material, and we have learnt about this double helical structure. We have learned few points. I'm going to just refer it. Then we will go ahead with this, with this diagram. What is in our friend? This is a chemical structure of DNA, deoxyribonucleic acid. It's the nucleic acid. Okay, and as you can see, it is very clearly, it's with yellow, it is written, no, nucleotide pair and or base pairs. What are the things present in our DNA? What are the things when we say? See, Watson and Crick gave the physical structure of it. That is, it double helical. This kind of structure is or was given by Watson and Crick. Okay, but the structure. This is the chemical structure. What you see. Okay, this is the chemical structure. What you have seen, and what all things we have learned last year only. I'm just revising it, and then we'll pull forward with this animation. Okay, now what are the things DNA made up of? Number one, it is it is made up of it is made up of sugar. Which sugar it is made up of? It is made up of pentose sugar. Okay, where is sugar molecule? You can see here. This is the sugar molecule. Okay, this is the sugar molecule. Okay, and the second thing is what is present is inside is the phosphate group. Okay, a phosphate group is present, and where is phosphate? Here, this is phosphate. Okay, after these two things, the third thing is a nitrogenous bases. In that also we have learned there are two types, purines and pyrimidines. Okay, okay. Now this, where is purines and pyrimidines? Purines and pyrimidines. These are If you see this, purines and pyrimidines are here. Of course, this what has been encircled here, this one. This are purines and pyrimidines. And this what we learned that there are two types, purines and pyrimidines, again adenine, guanine, cytosine, thymine. Okay, these are the four, and all purines and pyrimidines, and very much particular about their bounding. Adenine always make pairing with thymine, with how much of hydrogen bonds? You can see very clearly there how many hydrogen bonds they make with double hydrogen bond, two hydrogen bonds, while the guanine with cytosine, guanine, pardon, with triple hydrogen bonds. Okay, triple hydrogen bond. Okay, so in totality, if you see what are the things present in DNA, they have got a phosphate which forms a backbone. If you see, it is phosphate is known as its backbone. You see where they are present. See here. Okay, so they are known as forming a backbone, and then sugars, pentose sugar, and this phosphate group. Nitrogenous bases. So in total, that DNA consists of three things. Okay, you can proceed.
As a base pair. For example, E. coli has 4.6 multiplied by 10 raised to the power 6 base pairs, and bacteriophage lambda has 48,502 base pairs, while the haploid content of human DNA has 3.3 multiplied by 10 raised to the power 9 base pairs. It was in the year 1869 that Friedrich Miescher first identified DNA as an acidic material present in the nucleus and called it the nuclein. However, technical limitations made it very difficult to isolate such a long polymer intact, and therefore no further interpretations regarding the structure of DNA were made for several years. Finally, in 1953, James Watson and Francis Crick proposed a simple double helix model for the structure of DNA. They did this with the help from the X-ray diffraction data that was created by Maurice Wilkins and Rosalind Franklin. Further, James Watson and Francis Crick proposed that base pairing existed between two strands of polynucleotide chains, which was a distinctive attribute of their proposition. This proposition was based on the observations made by Erwin Chargaff, who found that the ratios between adenine and thymine and guanine and cytosine are constant and equal to 1 for a double-stranded DNA. In fact, due to base pairing, the polynucleotide chains possess a very unique property, that is, the strands of the base pairs are complementary to each other. Therefore, if we know the sequence of bases on one strand, it is possible to predict the sequence in the other. Moreover, when two strands of a parental DNA separate,
for this reason, this is the what you're seeing right now. The picture is the picture of this one. What you see right now is actually for replication. They separate both the strands. Okay, and why replications become easy in DNA is because of right now what we heard is because of the complementary pairing. We are very much we know that adenine always make pairing. Adenine makes pairing with thymine only, guanine with cytosine only, and that also guanine with cytosine with triple hydrogen bound, while adenine with thymine with double hydrogen bound. These kind of very particular nature of the nitrogenous bases make it easy for, from whenever replication time, whenever it happens. Okay, we'll proceed.
Each serves as a template for synthesis of a new daughter strand because of complementary base pairing, and the two double-stranded daughter DNA generated are identical to the parent DNA molecule. Thus, these discoveries provided a clearer picture to the genetic implications of the DNA structure, and soon the DNA double helix structure and its simplicity in explaining genetic implications became revolutionary. Let us now learn about the chemical structure of a polynucleotide chain present in the DNA double helix structure. The polynucleotide chain is made up of three components: a nitrogenous base, a phosphate group, and a pentose sugar, which is deoxyribose. Further, the nitrogenous base is of two types, purines and pyrimidines. The purines comprise adenine and guanine, while the pyrimidines comprise cytosine and thymine. Now, through an N-glycosidic linkage, a nitrogenous base is linked to the pentose sugar, forming a nucleoside, which can be guanosine or deoxyguanosine, adenosine or deoxyadenosine, uridine or deoxythymidine, and cytidine or deoxycytidine. Conversely, depending on the sugar present, a nucleotide or a deoxynucleotide is formed when a phosphate group is linked to
Okay, when we say nucleosides, nucleosides means, all this, there are three components. Out of all the three, if you minus sugar from this, it becomes nucleoside. Okay, when you add all this together, we call it, I'm just telling you the easiest way to remember what is the difference between nucleotides and nucleosides. The only difference is when we add all the three components together, we call it nucleotides. Okay, while when we minus these three components out of these three components, that is, what are the three components they have? That is nitrogenous bases, they have got sugar and phosphate group. Sorry, you have to remove phosphate group. When we remove phosphate group from the, I was right first, sorry, it is sugars only. Sugar, when we, no, phosphate group, when we remove, we call it as nucleosides. Okay, when we add sugar and all the three components, it is known as nucleotides. When phosphate is removed from the three components of the structure, when we remove and speak, I'm talking about, we cannot remove like that. I'm saying when, what is nucleoside? Out of the three components, minus phosphate is nucleoside. That is nitrogenous bases and sugars. From together we call it nucleosides. And when we speak about phosphate also in it, we say it is nucleotide. That is the only difference. Okay. Okay.
5' OH of a nucleoside through the phosphoester linkage. Also, a dinucleotide is formed when two nucleotides are linked through a 3', 5' phosphodiester linkage, and several such nucleotides can join similarly to form a polynucleotide chain. At one end of this chain is a free phosphate moiety at the 5' end of ribose sugar, which is called the 5' end of the polynucleotide chain. Similarly, at the other end of the chain is present a ribose with a free 3' OH group, which is called the 3' end of the polynucleotide chain. A salient feature of the double helix DNA structure is that it comprises two polynucleotide chains, the backbone of which
Okay, here 1 minute.
is called.
Okay, actually, just now what you heard is about the two strands, and both the strands are having antipolarity. If like this, they are saying here, this is 5 prime end, okay, this 5 prime end with a phosphate lying as free, right? Then here it is just opposite to it. Here you will see, this is, if this is 5 prime end, the other end of it here it is 3 prime, and it is what, it is having what, free sugar is free over there. Okay, now same way if you see the other strand, there it is antipolarity. This is, here it is 5 prime, then here it will be 3 prime end, okay, and here it will be 5 prime end, and just opposite to that is this side. Here it would be 3 prime end and 5 prime here. Okay, so this is antipolarity, and this antipolarity nature is also needed, which makes them, twinkled together. This is the reason that this kind of structure we get. This kind of structure we get, it's because of the antipolarity nature. Few features they are telling you, like just now they told you about and nucleosides, then they are telling you this, these are the features of DNA molecule, okay, and then about the antipolarity, they are telling you that both strands are having different polarity, which make them united together, and very stable structure they are. Yes.
Constituted by sugar phosphate with bases projecting inside. The double helix structure has several other salient features too. Its two polynucleotides chains have anti-parallel polarity. That is, if one chain has a polarity 5-3-, the other has 3-5-. Moreover, the bases of the two DNA strands are paired with the help of hydrogen bonds forming base pairs. Adenine is bonded with thymine from the opposite strand with 2 hydrogen bonds and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. Because of this structure, a purine always comes opposite to a pyrimidine, which results in a uniform distance being maintained between the two helix strands. Another salient feature of the helix structure is that the two chains are coiled in a right-handed fashion. Therefore, the pitch of the helix is 3.4 nanometers, and each turn consists of about 10 base pairs. As a result, the distance between a base pair and a helix is around 0.34 nanometers. Additionally, in a DNA double helix, the plane of one base pair stacks over the other, which, along with the
Okay, this point also, like since these nucleotides, very particularly, they including bonding with pyrimidines, and number of bonding also, like hydrogen bonds, it's always adenine with thymine with double, and guanine with cytosine with triple hydrogen bond. So the distance between them is also always same, and always same is particular. Then the correct distance always, because double and triple bonding, and the purines and pyrimidines. When we say the distance, we are talking about the structures are very much fixed, and the structure of bond and the structure of the molecules and all those things. Okay, the nitrogenous bases. So what happens? The distance is fixed, and that's why the distance, when we calculate between the two turns, the distance between the two turns, if you see in between the nucleotides, are 10 pairs of this nucleotide pairing we can find, and distance between each is 0.34. Okay, that is Armstrong's, and like this the calculation goes. And as you heard in the beginning of it, that in each individual, in each organism, the number of base pairs of DNA differs. Okay, in different organisms, it is different. It depends totally how many base pairs and all are there in it. Okay, yes.
hydrogen bonds makes the helical structure
And
very
only
stable.
0.1% makes that.
Stable DNA helix structure allows it to be the genetic material that has instructions for the development and functioning of all known living organisms. Did you know that the length of a DNA or deoxyribonucleic acid double helix in a typical mammalian cell is about 2.2 meters? This length is obtained by multiplying the total number of base pairs
topic
present
for,
in the DNA double.
The topic is over of DNA structure. What is DNA structure is over. Okay, if I conclude it in a very few statements, it is DNA is a double helix structure which consists of three components, that is a sugar, phosphate, and nitrogenous bases, purines and pyrimidines. And then further, each of it, they have got, like phosphodiesters, bond are formed, and then glycosidic bonds are formed between, as well as hydrogen bonds. These three types of bond are formed in this DNA structure. And then the next feature about it is both the strands are, having anti-polarity. Their polarity differences are there, and which makes them bond strong. And the like 5 prime to 3 prime or 3 prime to 5 prime, the polarity differs. And the next thing is complementary pairing between them, and the distance between the nitrogenous bases is always same and stable, always it is, as well as all these things are the, chemical features what you have seen, as well as the physical feature of DNA structure. Anybody want to ask anything on it? Else, we have to move on to the next topic, that is the packaging of DNA in the cell. Actually, a cell is, we know it's such a small structure, and DNA, if in a normal human being, the DNA, the length of the DNA, if we calculate, it comes 2.2 meters. Do you understand what is a meter? The cell which you cannot even see. Okay, inside it has got a nucleus. Inside that nucleus, when we say 2.2 meters of DNA is present, it is little unpalatable, right? So we will be learning how it is being done so miraculously inside each cell. Fine, but I wanted to know about DNA since it was last year's topic. I believe you have understood. If any doubts, kindly ask right now.
Ma'am,
Yes.
ma'am, can you share this video in the group?
Of course I will, and this I have already shared the video.
Is about 2.2 meters. This length is obtained by multiplying the total number of base pairs present in the DNA double helix with the distance between two consecutive base pairs. Now, the total number of base pairs present in a DNA double helix in a typical mammalian cell is 6.6 multiplied by 10 raised to the power 9, and the distance between two consecutive base pairs is 0.34 nanometers, or 0.34 multiplied by 10.
This is the calculation of how we are saying it is 2.2 meters. It's a calculation. As you can see here, they are telling you how it is calculated. The length of the DNA with total number of base pairs, will give you the, the 2.2 meters, what you can see. Okay, here it is given very clearly. The length of the DNA double helix is equal to total number of base pairs and distance between the two consecutive base pairs, and which we have learned that in a normal human being, one DNA has 6.6 into 10 to the power 6 base pairs, as well as the distance between them is 0.34 into 10 and to the power 9 meters. So when we multiply this figure, what we get is 2.2 meters. Yes, this is a calculation before we start that it is 2.2 meters. Yes.
to the power minus 9 meters. By multiplying these two factors, we get the length of the DNA double helix as 2.2 meters. Have you wondered how such a long polymer is packaged within a typical nucleus with a dimension of about 10 raised to the power minus 6 meters? To understand this, let us learn about the packaging of a DNA double helix, which is different for both prokaryotes and eukaryotes. In prokaryotes such as E. coli, the nucleus is not very well defined. However, the negatively charged DNA is not scattered throughout the cell, but is arranged in large loops, is held together by a few positively charged proteins in a region known as the nucleoid. On the other hand, in eukaryotes, the manner in which a DNA double helix is packaged within
This was about the prokaryotes. Okay? The prokaryotes, what you saw just now, the figure, it is a little reminded. I can show that structure if you can hold on. Yes, this structure, little zoom it and we can see, yes, this is DNA. And what you can now, we know that in a what is the difference between prokaryote and a eukaryote? We know the basic difference in prokaryote and eukaryote is, in a prokaryote we don't find a definite nuclear membrane. There is no definite nuclear membrane. Okay, so and you can see how they lie. They lie inside the cell, the cytoplasm in this form. I'm talking about DNA. Okay, now here this is the structure of DNA, and when you see this, okay, this structures, you can see this, they form like a flower. Okay, here this flower like structure, what happens here is, okay, this what yellow colored thing what you see now is actually positively charged. Okay, and these things, this what you see, this loop like things are actually DNA which is negatively charged. These are the proteins. Okay, and these are the DNA. What they do, they on this nicely they loop